We hardly hear you.
Okay, now is it better?
Yeah, better.
Okay, so I know that Leon has a lot of material to cover today, so I'll do it briefly. As you will see, Leon's research is very interdisciplinary, and that's been the case since he started his academic career, basically. And he did some of his work in the biology department, and the PhD was in the computer science department at the Technion. Leon is currently a research fellow and the academic director of the machine learning and data science program in our school at IDC. We're very happy about that, and I'm very happy to host Leon here today. And I should also say that Leon is a father of two, and that he was born and raised in Ota Kikar, deep in the Israeli South, which is something you may not have known about Leon. And with that, Leon, please, thank you.
Okay. So thank you, Zohar, for the very brief introduction so we can have enough time to cover all the material you have. Let me just start by sharing the slideshow. This is it. Yeah. Okay, yeah. So, hi everyone, and my name is Leon, as Zohar introduced you, and I'm going to talk to you today about DNA-based data storage systems, which is a bit of an odd topic, let's say, but I hope interesting, and I hope you'll find it as exciting as we do. And I'm gonna try and focus on the more computational aspects of building such a DNA-based storage, and describe a few projects that we did regarding this topic. Just briefly about myself. So as Zohar said, I come from a very interdisciplinary background. My background spans statistics and machine learning, then maybe standard computer science, let's say, and the aspects of molecular biology and cell biology, where I am interested in evolution and in the systems biology, and specifically in synthetic biology. And I think that the intersection of those very diverse fields brings very interesting projects and studies. One of them would be studying the coding properties of DNA-based storage, which will be the focus of today's talk. So just briefly, we're going to start by a very, very high-level introduction of what is synthetic DNA and how can we use it to store data. Then we'll describe 3 projects. The first one would be a project I did well, all of them are from my PhD at the Technion, but the first one would be, let's just dive into the project and you'll see yourself. Okay, so DNA, what is it and what do we do with it? We all know DNA, we all have it. All organisms use it as almost all organisms use it as the genetic material. After the last year, we all know RNA viruses, so not all organisms, but almost all organisms. It starts from the very basic and small organisms and goes up to the most advanced organisms we can think of. We all have DNA in our cells that contains the genetic information. Now, what is the role of DNA, or the general role of DNA in nature? So the common way to view it is as if DNA is the hard drive, hint for the next part of the talk, where the genetic information is stored, and then it can be replicated, very much like we copy and paste files in our system and in any digital system, and then it can also be transcribed into RNA molecules, which are more, let's say, functional molecules, and can be translated into proteins which are the actual, let's say, workers in our cells and bodies. And this is a very, very, very high level view of what the role of DNA in nature is. The truth is that the picture is much more complex, and we're not going to go anywhere near what's really happening. And the next thing would be that we're not talking about DNA in nature in this talk. We're actually talking about synthetic DNA, and we'll get to it in a few slides. So let's start with a small question. Do any of you want to guess what we see in this picture?
An old storage device.
Yes. So this is indeed an old storage device. This is a 5 megabyte hard drive built in the 50s by IBM. So we've come a long way since this was the way we stored information. But actually, it seems like we're a bit stuck. So again, after the last year, I don't have to explain the notion of exponential growth, and specifically not to this audience. But if you see in this slide, you see the demand for digital information storage needs worldwide, as it is growing in the last decades. That's it. And we're approaching the limits of what we can support using the conventional storage devices. So DNA, as we said, was used in nature as the material used to store DNA, to store information in, and therefore it is a very appealing candidate to maybe be used as an alternative storage media. If we compare the properties of DNA as a storage device in nature to the ones of the state-of-the-art magnetic tapes-based storage devices, we see that in terms of the physical density, how much information can we actually store in every, let's say, square millimeter? We have DNA which is much better. When I say much better, is 12 orders of magnitude more dense than what we see in the state-of-the-art tapes. And if we look at the robustness or the lifetime of the storage media, while standard magnetic tapes or CD-ROMs, whatever you can think of as your storage device, has to be rewritten and replaced every few years to maybe few decades, DNA can survive in nature for centuries, even up to 1 million years. So also, and be readable. Okay, so we can retrieve it and read the actual information stored in this DNA, natural information. So in both terms of physical density and robustness, DNA is a very appealing candidate. The last point would be that DNA will never go obsolete. So while back in Otakikal, in the desert, my parents still have VCR tapes of me doing my first steps, or me reading the Torah in my bar mitzvah or whatever. And the problem is that we have no VCR machine that's able to read it. So those tapes are useless. We cannot retrieve the information from those tapes. And this would never happen to DNA. So as long as we're here and we have DNA in our cells that we want to read for other purposes, for example, medical purposes, for say, we'll be able to read the content of a DNA molecule. And this is another aspect of this is that it's the same DNA molecule that is being used to store information, genetic information, for the last, let's say, 3 billion years. And then the machinery in the cell that was developed during those 3 billion years is very, very efficient and was optimized by evolution to be very efficient, which is not the case in standard media or storage media. Okay, so let's say I convinced you that DNA is a very appealing candidate for storing information. How do we do it? We start up with just a file, okay, as you see here at the left of the slide, just a binary file. Then we somehow have to encode it into a DNA sequence. This would be the majority of our talk today, probably. Once we have a DNA sequence that contains the information that we want, we need to write it. We need to actually do the physical writing procedure. Imagine the actual flipping of the bit in the hard drive. So we have to actually somehow write this physically. In DNA, it means to synthesize the DNA molecules that contain the sequences that we want. Then we can store it. Let's say if I can have this. Yes, okay. Then we can store it for as long as we want. Just put it in the fridge in our lab and wait for the right moment. And then when we want to actually read it, we can use the standard DNA sequencing machinery that we have in every research facility, in every hospital today, that can take the molecules from the fridge and return a set of sequences that contain that is the content of those molecules. Once we have the sequences, the DNA sequences, we can decode it back into the binary file that we have. Okay, so yeah, sure.
So you said it should be in the fridge, not like in nature. So it's still a minor limitation until it can be in bones and so on.
Yeah, so there are various ways to store DNA. We can have a whole talk only on the different physical ways to store the DNA so it will last for longer. One way would be to freeze it. The other way would be to just dry it and then keep it. When we order the DNA from the company, we don't get it as liquid, we get it as a powder, which does not have to be frozen. And then only after we add it into water, then we have to keep it in the fridge. There are other ways. We can maybe pack it into small silica balls so it will be contained even longer. But there are limitations here. I think the main issue here is the fact that there's still the physical aspect of storing the actual molecules somewhere and be able to manually or hopefully automatically retrieve those molecules and continue the process of sequencing and decoding. Anat, yes?
Maybe you said and I missed it, but can you repeat again what kind of information the best way to store it is by DNA, like in the real life?
Yeah, okay. I don't think I have discussed it. I don't think I did. I discussed it. So let's say, since the technology for writing DNA and reading DNA is very high throughput, but very low latency. Oh, sorry, high latency. Okay, so it takes a lot of time, but eventually you get a lot of data together. So you wouldn't want to replace your USB stick because it will take you hours or days just to write one file and read it back. But if you have a very large archive that you know that you want to store for decades, and you want to be very sure about it being stored correctly and being able to retrieve it somewhere in the future, then DNA is...
maybe if I want to start a new religion and someone wants to write a new Bible for 2,000 years in the future, I'm asking really, I mean, what kind of,
yes, so
which scenarios are we talking about except starting a new religion?
So if we look at the actual scenarios that are now trying to be pursued by like large companies in the field, let's say Amazon or Microsoft, that are interested in this technology, so they're looking at as the deep freeze archiving solution. If you have a lot of data that you want to store for a lot of time, but you don't really care about the retrieval time. It's okay if it will take them 1 week to retrieve the information for you, then it is very efficient to store it in DNA. We did not discuss the efficiency in terms of energy consumptions because we simply don't have time, but it's also very efficient in terms of energy consumptions because it's only a tube with DNA that is stored somewhere, and you don't have to maintain a whole computer that actually store it. Okay, so I think this covers this. Okay.
Thank you.
The very basic encoding scheme would be, the very naive encoding scheme would be, since DNA is a polymer that consists of 4 building blocks, 4 letters. So we have a 4-letter alphabet. Let's take every 2 bits. Keep a dictionary that translates every 2 bits into 1 letter of DNA. And then whenever I get the binary message, I can simply translate it every 2 bits to 1 letter and synthesize the resulting DNA sequence, and we're good to go. Okay, this would be the very basic encoding scheme for DNA storage. Now, a little bit about the technology for actually synthesizing DNA. So we're talking about high throughput synthesis of oligolibraries. Oligolibraries would be a set of a lot of DNA sequences that are being synthesized together. The main idea is that you get a set of sequences. You have different vendors and different technologies, but they all have the same basic principles behind them. And eventually, after this process, you'll end up with a tube containing all your DNA molecules. Now, hopefully, the next movie will work. Let's see if it happens. Let's see what happens. Yes. So basically, what happens in DNA synthesis, it works very much like inkjet printing. So you have a cartridge with 4 heads containing the 4 DNA bases that you can have. And cycle by cycle, you just inject the next letter that you want to let's have it again, that you want to synthesize or you want to elongate your molecule with, and then it connects to the previous strand that you already have, and eventually, after all those cycles, you can cleave your molecules from the surface, and you get what I love to call the soup, a soup containing all your molecules, all the strands that are DNA strands containing the sequence that you designed. Now, I did not discuss any errors in this process. Let's, for now, assume that this process is perfect. Okay. Wait, what happened? Okay. Okay, so what are the limitations or the properties of this technology? Basically, since it is an iterative process and each cycle has some error probability, we're limited in the number of cycles that we can run. So this actually limits the length of the molecule that we can synthesize. On the other hand, as I said already, this is a very high throughput process. So we can do the same synthesis process in parallel for up to 1 million or even more different DNA sequences. And another level of maybe multiplicity or parallelism is that every sequence that we synthesize, we get in many identical molecules and many identical copies of the same sequence. So this, we'll call it maybe an inherent redundancy in the process. And the last property is that we end up with only 1 tube containing this very mixed population of molecules. Okay. So far, so good.
One question.
Yes.
Is there any limit on the length that you can prepare? I mean, if you have a very long file and you have to store it, you store it in multiple sequences, or
Yes.
what?
Yes, yes. So we are limited by the length of each one of the sequences for about 200 or 300 cycles, so 200 or 300 letters of DNA that can be contained in one sequence. And then if we get a very long file, we just have to break it down into pieces, and each one of the pieces will be translated into a short enough sequence that we can synthesize, and then we move on. Okay.
But Leon, this means that you need to also preserve the order.
Correct.
And unlike arrays of
correct.
register, it's very hard.
Yeah, Guy, you're correct. Since we say that we break the file down and we also mix all the parts together. So what we actually do is we preserve the beginning of the sequence to be the address. Okay, so now every sequence that I read, I can locate the address of it using what we call the barcode. And then the rest of it would be the payload, the actual information. You're right.
Okay.
Question, why do we have so many identical sequences?
This is just a limitation of how the chemistry works. And you're pinpointing the actual property that we would like to take advantage of. You'll see it in the next slide. Okay, so let's define two densities, let's call it, properties of DNA storage system. The first one would be the logical density. How many bits can we write in every cycle of the synthesis? This is actually the property that affects the overall cost of the system the most. And the second one would be the physical density, which means how many bits can we store in every gram of DNA, which is just to give you an idea, it's roughly in the hundreds of petabytes per gram. We'll see a visualization of it soon. Okay, but the logical density is limited to two bits per synthesis cycle, or 2 bits per letter, DNA letter, let's say. But what if we had 8 DNA letters? Let's keep it in mind for another slide. Okay, this is an overview of the process. I think we covered it, so we don't have to go over it again. In the previous decade, almost a decade now, we've seen numerous studies. I only put here the ones that actually did DNA synthesis in the study. There are many theoretical studies that we are not included here that implemented different encodings and different tackled different questions and stored information on roughly 1 megabyte scale. Maybe one work that stand out here is the one from Organic et al. It's a collaboration with Microsoft Research, where they actually started going above the megabyte scale and go into the gigabyte scale. They already reported doing, I think, almost 1 gigabyte, the last report they had. Okay, I promised you a visualization of how dense this is. So basically, all those studies we saw work around 20 petabytes per gram, which is, you can take 1 teaspoon of DNA and potentially store all the YouTube repertoire collection of movies in this 1 teaspoon of DNA. Or alternatively, you can take 1 truck fully loaded with DNA molecules, and you get all the digital information we have in the world. This is a lot more dense than what we see in actual storage.
There's also one more limitation, which is the writing time. So how long would it take to actually write all YouTube videos on a DNA?
Yes, okay, so
that would take years, no?
Not only the writing time, since you're right, there are not enough DNA synthesizers to do it, but it also actually, okay, so now I want one specific movie. I have to somehow dig into this spoon and take the 1 grain that I want and go and sequence it. It's not very practical yet, but this just gives you the idea of what is the potential, because we know to store in this density, not in this scale yet. Okay. Okay, so now after this very long introduction, let's dig into our work. This is the work we did jointly with the lab of Roy Amit in the Technion, and was published last year, or maybe we're 2021 now, so last year. And this actually answers the question, so what if we had 8 DNA letters? So allow me to introduce you 2 new DNA letters, M and K. And I'll describe them as M is simply a mixture of A and C, and K is simply a mixture of G and T. And maybe seeing the actual synthesis would be the best example of what is this mixture we refer to. So if you see here in this same video describing the synthesis process, you have the 4 standard DNA cartridges in the inkjet printer, and you also have two more cartridges that contain pre-mixed solution of G's and T's or pre-mixed solution of A's and C's. Now let's see what happens when we actually run the synthesis. When I just want to put A, I put A, but whenever I want to put M, for example, I put a mix of A's and C's, and then half of the molecules that resulted of this synthesis cycle are A's, and half of them are C's. Okay, in this same position number 3 in the sequence, I have 50% A's and 50% C's. This is where we actually take advantage of this inherent multiplicity that Anna earlier asked about. Okay, so if we now collect a sample of the soup of molecules that we have and look what happens in this, for example, the 4th position, we'll see a mixture of A and C, and then we can infer that this was actually M. This was a letter that we designed it to be a mixture of A and C, so we can infer it as M. Okay, so we did the same process as earlier. We synthesized, we then sequenced using the same sequencing machinery. Just after we sequenced, we had to infer the ratio of the different bases in order to identify the composite letter. We call those mixture letters composite letters. And if we actually had 8 letters, we could store 3 bits in every letter in our alphabet. Okay, so this increases the logical density.
But actually, you can store even more than 3. I mean, it's not only half and half. You could take, I don't know.
Yes, you're asking about my next slide. So let's wait for the next slide. So actually, we designed it to be M. So we have a very big pool of molecules containing a 1-to-1 ratio of A's and C's. Now we get a sample of it. Let's say we only read 20 molecules, and we see 8 times A and 12 times C. Now we have to decide, is this representing an A? Is it representing an M? Or is it representing a C? Okay, this is being repeated over and over. And now we can ask, so what is the distribution of those observed set of reads that we have for each one of the letters? What would it be?
Probably N.
So this is actually a multinomial distribution where we read N copies, and the original probabilities are defined by the design letter that we wanted. So if we design the letter which is 50% A and 50% C, for example, M, so we'll have a probability vector which is 0.5, 0.5, 0, 0 over A, C, G's and T's. And this would be the probability vector or the parameter for the multinomial distribution of the set of N observed reads. Was this clear? I hope. So now,
question. Sorry.
Yeah.
The density slide that you had with the sugar, does that take into account all this duplication, or does that assume that each sequence is unique with its own unique data?
Oh, so this takes into account the actual physical density, which takes into account the duplication. Yes.
Sorry, another question.
Yes.
You said that the multiple molecules helps in this synthesized nucleotides, but why it contributes to this?
Because the only way we can actually get a mixture of A's and C's is if we do synthesize more than 1 copy of the same sequence. Since we synthesize 100,000 copies of the sequence, we can plan it as such that we'll have 50% of them with A and 50% of them with C. If we didn't have this multiplicity, we couldn't do it.
Okay.
Also, Leon, can you hear me?
Yes.
Yeah. Also, right, it also comes from, you need to synthesize more than one just to be able to correct for errors and things like that.
Correct,
so
yes.
it's not just
some of the redundancy is used for error correction, indeed. Simple error correction usually. Okay, so we have to come up with some mapping functions that will get the observed set of reads, and we'll map it into one of the letters in our alphabet. And as Toki said earlier, I think, it doesn't have to be an alphabet of what we call resolution 2, only 50% and 50%. It could be an alphabet with higher resolutions, which will only make the mapping maybe more complicated for us, because then the difference between 2 neighboring letters would be more subtle, let's say. Okay, 2 ways to do this mapping would be, the first one would be to actually look at the frequencies of the observed letters in the set of reads, and look for the closest probability vector in the alphabet that we have. Another one would be to use the KL divergence function, which will be the KL divergence between the observed set of reads, or the frequencies, the observed set of frequencies, and each one of the probability vectors of the letters in the alphabet. It turns out that using the KL divergence is actually equivalent for doing the maximum likelihood estimation of the letter. Or choosing the letter that maximizes the likelihood. Now let's see what we'll not go over all the derivation here. We just see that if we define the likelihood, the log likelihood of the multinomial distribution as such, and remember n comes from the observations and p comes from the letter, and we do some simple derivation of the formula for the KL divergence, we'll end up with the fact that the KL divergence is just a constant that only depends on what we see, minus the same likelihood term that we saw earlier, divided by some constant term, okay? So actually, minimizing the KL divergence would be equivalent to maximizing the term of the log likelihood that we see here, okay? It's nice.
So the likelihood is independent by the amount of repetition that you have in your soup?
No, it is dependent, but okay, so choosing the right one only depends on the ratios. It does not depend on the actual n, capital N that you see here. You're right. So the maximum likelihood one, or the closest letter, would only depend on the actual of the frequencies, and we don't care about the actual the sum of all the observations. You're right.
But it's counterintuitive, no?
No, okay. It's not. It will be easier to infer as you have more if you have more observations. It will also, but the frequency. But if you get the same frequency from 100 samples or the same frequency from 10 samples, you'll choose the same one. Okay, you'll choose the same method.
But you not really get the same frequency. You have some distribution to get that.
Yeah, okay. The probability to getting the frequency depends on the size of the observation set. You're right. And the larger it is, the higher the probability to be in the let's call it the range where you infer correctly. You're right. I had a slide of it over it, but we simply don't have time for it. So what we see here is that we actually increased the logical density because we extended the alphabet, but we decreased the physical density somewhat because we now need to use this multiplicity. We need to have more molecules. We have to read more molecules in order to actually store or read every one of the letters. Okay. Let's do a very quick wrap up of the errors, and we'll run through the results to see at least one more project. Okay, so we can have errors in DNA synthesis, which can be just substitution of the wrong letter that we see, or can also be dropouts where the entire molecule is just not observed, simply because we just did not sample it in the sequencing. Now, there are other people here that can explain this much better than I do, I guess, but let's do a very quick overview about what happens when we want to maybe communicate information over a noisy channel. So what we basically do is we take our information and put it through an encoder, which translates it into a code word C, which is then being communicated over the noisy channel, which introduces errors. And then a decoder can hopefully retrieve the original letter U, or U hat, that will be hopefully identical to the original message U. The one thing to remember from this slide is that in order to do this, the encoder actually has to add redundancy. So we send a longer message in order to be able to correct some of the errors that we have in the message. And choosing how to maybe do the translation from the original message to the code word C takes into account different parameters, which we will not get into. You just have to remember that you have to keep in mind what is the technology that you're working over. So you have to make sure that the actual encoding scheme supports the errors that your channel introduces. Okay. In our case, let's skip this. We use different error correction schemes. And now let's briefly discuss our results. So we actually took a 6 megabyte file containing the Bible, translated it into just 6-letter composite alphabet. 6 letters because we only had the option to add 2 cartridges to the machine that does the synthesis. And by this, we increased the logical density by 25%. Okay? And we maintained a physical density which is comparable to what we saw in other studies. We used Reed-Solomon encoding or Reed-Solomon error corrections directly over the composite alphabet. So if the alphabet has 6 letters, we used Reed-Solomon over GF7 to the third. We're not going to get into it. If anyone is interested, we can discuss the actual bits and bytes of this encoding and error correction in details offline. And let's see some results. So we synthesized more than 1 million oligos, or more than 1 million different DNA sequences. Each one of them was about 150 nucleotides long. And this means we actually saw many millions of positions where we designed them to be M. And the distribution of what we actually observed, or the ratio to A, the A to C ratio that we observed in each one of these positions is displayed here. You can see that A, the center is not directly over 1-to-1 ratio. And B, there are a substantial amount of reads that are, or of not reads, of positions where the observed ratio is not inferable or directly inferable into being a 1-to-1 ratio if we use a higher resolution. Okay. Another property of what we saw is that the success probability is highly dependent on the sampling rate. So if we only sampled a very small subset of the reads that we have, the success probability drops substantially. Okay. And let's summarize this project. We actually used physical density comparable to others. We had some error correction challenges that we had to overcome, which I will be happy to discuss in details. And then we saw how the composite DNA extended the alphabet and increased the logical density. I did not show it, but we also showed how this can potentially reduce the cost by as much as 50% in some theoretical scenarios. Okay, now I'm going to briefly describe, hopefully not so briefly describe, a project that is led by Inbal Preuss, who is here also, a great master students from here, from IDC. And it starts by reminding ourselves what are the drawbacks of using the composite DNA. And the main drawback is that we have to actually infer the correct ratio between the 4 DNA bases in each one of the letters. So when we observe a letter, we have to infer the correct ratio. This in turn requires high sequencing depth and requires the synthesis technology to be very, very precise to generate these actual specific ratios. And we overcome all those drawbacks with an approach that contains 2 main, let's say, properties. The first one would be to synthesize DNA in shortmers, and I will describe it in a second. And the second one would be to use what we call a combinatorial encoding, which does not require the actual inference of the ratio between the different building blocks. So what is shortmer synthesis? We define a set of DNA k-mers. So let's say trimers, it's just 3 letters of DNA glued together. Okay, so we have a set omega containing n different DNA k-mers. We can define it such that it has a minimal distance d between every 2 DNA k-mers, and this also ensures low probability of mixing or mixing between 2 different k-mers when we observe one read, because one error would not get me into another, let's say, legal k-mer. Okay, we use those k-mers as the actual building blocks of the synthesis, and let's see in Bal's beautiful video that shows it. You see here. So you see that the actual cartridge is much larger now. It has the 4 standard DNA letters, but also many k-mers. I think in this case it's 10 or 16 k-mers, 3 mers in this case. And then whenever we want to synthesize a mixture of k-mers, what we'll do is we'll take the 3 k-mers that we want to synthesize together. We'll mix them together. We'll see in a second. Now we added the first one, we added the second one, and then we can mix them together. And what happens now is that this synthesis cycle will do first a mixture of the k-mers, but also each one of the sequences that or the molecules that we synthesize will be elongated by 3 bases together that came together from this 3-mer. Okay, this is the idea behind short-mer synthesis, or even mixed short-mer synthesis. We synthesize in short-mers, which are the building blocks, and we mix them together just like we mixed earlier. Okay, now when we observe a read or set of reads, we look at the specific position, we look at the k-mers, the 3-mers that we observe, and all we need to do is to infer which from the 16 original 3-mers are the 3 that were included in the mix in this position. You can also see here that even if we have one sequencing error, it does not affect the inference, because in this case, the distance between every 2 legal 3-mers is 2. So one mismatch would not get me into another of the 16 k-mers, and this read will simply be disregarded. Okay, so from this set of reads, I can infer the binary vector, in this case of size 16, that says 1s in the 3 positions where the 3 3-mers that were included in the subset that is the letter that we want. In this case, sigma 39 is defined as the subset that contains x1, x4, and x16. Okay? Now, our alphabet is basically a set of subsets of omega. Or, yeah, a set of subsets of omega. We can use the power set, only minus the empty set, but the power set. So we get 2 to the n minus 1 letters in our alphabet. However, this is, while very dense or very large alphabet that we get, this is hard to decode because we don't know if, let's say, we've seen 4 k-mers in the letter, and we don't know if we're waiting to see the 5th one, or we can wait and decide that this is the letters that are included in this subset or in this letter. So a different approach would be to use only the k power set, which means all the subsets of omega that have exactly k elements in them. Okay, this way the size of the alphabet is n choose k. I think I have to repeat the basic idea here again because I did it very quickly. So we have a set of building blocks, and every letter in our alphabet would be a subset of these building blocks. If we, in this case, we only define as legal letters or OK letters that included in our alphabet, the subset that contains exactly K different building blocks. Now, if we want to identify the subset that we're looking at, so we get a set of reads, and in a specific position, we get the distribution of the different over the different elements in our omega, different k-mers. And now we can define a rule that says if you've seen at least m reads for exactly k different k-mers, because we know we're expecting to see exactly k-mers, then you can stop, and this is the subset that you're looking at. Some of you may see that if we define m to be 1, and we're waiting to see at least one read of each one of the k different k-mers, this is actually the coupon collector distribution, which is a sum of geometric random variables. It doesn't really matter, but we can say that the expected number of reads we have to read in order to observe at least one representative of each one of the k different k-mers is roughly k log k. Okay, so this would be the binomial encoding. A lot of details about how we can encode with this. We can summarize it by showing an example of two different alphabets. Let's focus on the binomial one. So the one that we've seen with n choose k. So if we use 16 choose 7 alphabet, we can increase the density even into taking into account all the error correction overhead that we have to take to be comparable to other work. We have a fold increase of almost 6.5 fold increase in the logical density. Okay.
Compared to what? What's your
compared
baseline?
to the standard DNA? Yes, compared to standard DNA. You're right. While what we had in our composite DNA was 25% increase in the experiment that we did. Okay, so here is a 6.5 fold increase.
And are you? You said that you're waiting for one read for each k-mer, right? But that's not very robust.
Yeah,
like if there
you're
are
right.
errors, then you're going to read the
Yeah,
wrong.
so you're exactly describing the next direction section in this slide. The next direction would be to maybe see more rigorously what is the required number of reads in order to get the probability, the error probability lower than alpha when we require m and explore different inference algorithm or approaches. So you're correct, and this is exactly one of the directions that we want to take it to do some more analysis on the different inference methods. Another thing we want to do is to do a small proof of concept experiment where we're going to probably do it with a lab in Bar Ilan, the lab of Ayala Hendel in Bar Ilan. I have 1 minute, so I'll take like 3, okay, and describe in very, very broad lines the project that we have together with Professor Eitan Yakovi from the Technion, his student Omer, and from our side is Roy Shafir, who is the lead researcher on this project. This actually talks about enzymatic DNA synthesis, where we replace all the inkjet printer synthesis we showed so far with synthesis which is based on enzymes. And we all know that enzymes look roughly like this. Okay, so enzymatic DNA synthesis or the approach that we're dealing here goes as such. You have the same beginning of a molecule that you start off with, and then you have 2 enzymes. One that is Bob the Builder, that actually tries to take all the substrates, all the DNA molecule, all the DNA nucleotides, and just connect them into the elongating molecule. While the other one would be Pac-Man, that tries to eat all the DNA, all the nucleotides in the soup as fast as it can. Okay, and the question here is the competition between those 2 enzymes, and you end up with a red tail in some length, which is unknown, or random variable, the length of the red tail. It depends on the rate that one of them is connecting them, and the other is eating them or degrading them. Okay. So the general idea was to this actually what it causes is what we call the starter synthesis channel. So you start up with I want to put an A, and you end up with a series of A's with a variable length. Okay, and the goal here is to get a set of stuttery reads, split them into the consecutive sequences of the same letter, and then each one of those we should infer what was the original designed length. Did you want to put one A? Did you want to put two A's and we got this stutter, and so on. Okay, I will not get into the details. I will just say that we modeled it as a negative binomial distribution that depends on the probability to stop stuttering and the design length K. And using maximum likelihood reconstruction algorithm actually takes the observed reads, and the goal is to output K, or the design length K, which is the most probable, or with the highest likelihood to produce this set of reads. This depends on two parameters. The first one will be the prior probability to see each one of the Ks, and the second one will be the probability to observe a set of reads given a specific K. And some results we can see. I will not get into the details, but we can see that this maximum likelihood reconstruction is much more efficient than other approaches, and we get much lower error rates. I think this covers the main idea of the enzymatic stutter channel. I hope the main idea got through. And if not, I'm happy to describe it in more details, or even direct you into Roy, which will probably do much better work than I do. And the next direction would be to maybe support a more complex error channel, not just stutter, but on maybe the mixture of stutter and deletions and substitutions to be more realistic, and benchmark it over the actual results that we have from companies that try to implement enzymatic synthesis to see how well our results are behave on their data. And by that, I want to end and thank first Zohar, who's my mentor and my PhD advisor for, I don't know, 5 years and now a very great colleague in my work as the director of the machine learning program, and the rest of the Yechini group, which are fun people and very smart people to work with. Always happy to have valuable discussion with. And the labs that we collaborated with, the lab of Roy Amit, the lab of Ayal Hendel, the lab of the research group of Eitan Yacobi, Twist Biosciences, who's the company that does the DNA synthesis, who worked very closely together with us on the composite DNA synthesis, and the Adams Fellowship, who supported me through my my PhD in the last 4 years. And by that, I want to thank you for listening, and I will be happy to take any more questions that you might have.
Great. Can you hear me? Wait, can you hear me?
No, you'll have to come here if you want, or I'll have to mute myself if you want.
I have a question.
Yes.
I was wondering, I tried to understand the motivation behind the enzymatic stutter mechanism. So why are you interested in synthesizing DNA not using a machine?
Yeah, so I would mainly look at the last bullet here, and I would say that vastly, biology is just better than chemistry in doing stuff. Okay, because biology is optimized for doing stuff. we have enzymes that are optimized to connect DNA nucleotide one after the other. It just happens much faster than what happens in any machinery that we can build ourselves, or robotics, or whatever we build. We will never be as efficient as what nature does. We'll have to understand how we can adjust our needs so that nature can actually help us. So the main answer for this would be that since evolution took care of optimizing the machinery for us, the machinery is much more efficient, and then we would want to use this instead of the actual chemical-based synthesis.
Yeah, the claim is that it's going to be cheaper. Currently, there is no company that does that on any scale which is worth mentioning. Okay?
Yeah. But I guess the advantage here is that you're willing to be flexible with what you actually synthesize. You can ask things that are easier to synthesize using an enzyme.
Yes, yes. So maybe this even takes me to the actual naive approach to deal with this stuttering noise would be to just not put the same letter twice in a row. And then you only have like a 3 letters alphabet, and you're okay. You just need to wait for the switch, and this is the encoding of the information. But we did not want to pay the price of going down from 4 letters to 3 letters. There are some ways in between when you can, let's say we're only allowing 1 letter or 5 letters or 10 letters. So it's easier to distinguish the length of the stuttering, for example. So there are ways in between, but you're right. We'll have to adjust our requirements from the synthesis, since this is used for storage, and we're able to define what it is that we want to synthesize. We can define it such that the machinery will do it well enough.
That's nice.
I have a question.
Yes, sure.
So in binary, in electricity, we have 2 letters, 0 and 1. In DNA, we have 4 letters. Are there any beginnings of works with something in nature that have more than 4 characters?
Yes, so if you look at flash drives, for example, flash drive doesn't have just 2 letters in each one of the components. It has 8 voltage levels, and the encoding is the voltage level. So you have 8 levels that you can distinguish between and use them as your.
But I think they are encoded as 8 different binary levels. So the question is whether there is in nature.
So the answer is yes. There are people working on adding a chemical 3rd pair of letters. So it needs to have also the properties of DNA that they pair with each other. You didn't talk about that, but DNA, A binds to T and C binds to G. So people are working on chemistry that will add a 3rd pair like that. Okay, but I don't know if I would call that nature. It's chemistry. Okay, but
actually, can you take your tools and use your tools similar levels to move from any K alphabet from so?
Yeah, so if you're asking about the more theoretical aspect, so there are some studies that we've seen that trying to maybe define those more higher alphabet channels of information, but not many, and most of them focus on many specific questions because you have to base your theoretical questions on some real world application. And I think this was one of the first attempts to actually come up with a real world motivation for those complex large alphabet stuff. Okay.
I also have a question.
Yes.
You presented the combinatorial scale for encoding.
Yes.
And I wonder if you look at the variable length encoding. Potentially you can, win much more if you allow for that. So K is not fixed anymore. Okay, basically.
K, the K-mer K.
Yeah.
Okay. Yeah. So for technical reasons here, because pre-glue the three bases into one building block. Okay, so we can generate a set omega that contains both 2 mers, 3 mers, 4 mers, and 5 mers, and choose which one of them to use, such that the overall length would be minimal. That's one approach. The thing is that in this, at least in the synthesis approach that we're looking at, we're paying per cycle. We don't really care if we add a 2 mer or a 10 mer in this cycle. We're paying for the cycle. So,
right.
so
you can, but you can represent data in a much more dense way. So you can win a lot with compression in this way. You can get near entropy, basically.
Yeah.
Right?
So this goes to the work of Huffman coding, where actually this was the first thing we did in my PhD maybe 5 years ago, where we tried to encode in variable length DNA, and eventually we dropped this direction. But I agree that there are interesting questions to ask about variable length DNA sequences.
But in this case, you can compress using Lempel-Ziv and then encode the results.
It's first compressed. The data is always first.
Yes.
Yeah.
But I have another question related to COVID. Can you encode the information using RNA?
Okay. Let's start the discussion about RNA. The problem is RNA is much less robust. So if you just put RNA in a tube, it will degrade itself. Any RNA molecule has like this property that if you let it sit for a while, it will degrade itself.
You need a better refrigerator, -70.
Yes, exactly. You see, you have to, because it is an RNA-based vaccine, you have to keep it in -70.
But it's a theoretical question, yes. I mean, in principle, I mean, is it better?
So in principle, I would guess, and if we're going again to the maybe the equivalence of computer software or computer machines that we were used to, I would call the DNA the hard drive, and maybe use RNA as the cache or the caching machinery. So if I want to access a specific file, I can transcribe it into RNA. It will be available for me for the next few hours, and for easier reading. And I cannot change it because I cannot change the original file in the hard drive this way. But it's maybe easier to read. There are some sci-fi approaches that try even to go up to proteins and say, okay, so instead of we'll write the information on DNA, we'll translate it into proteins, and then we'll read the proteins, which is much easier to read in some scenarios. So the question is, it's a good question, and we just have to find what is the right goal for RNA in this big picture.
So Udi charged me with keeping the time.
Yeah. You don't want to know your bill.
Exactly. So and Leon, of course, is around.
Yeah, I'm around.
You're all welcome to talk to Leon about all these interesting questions. And thank you very much. Thanks again to Leon.
Thank you all for coming.